Very good morning. As part of lecture series on cholangiocytes, I will be presenting on cholangiocyte crosstalk with innate immune cells. So this topic on immune cells is pretty much complex, but I will try my best to keep it very simple so that everybody can understand. So during my presentation, this will be the outline, the cholangiocytes as immune sentinels of liver, their mechanisms, how they crosstalk with other immune cells, the clinical relevance in various liver diseases, how bile also drives the modulation of cholangiocyte immune responses, some of the ILBS data, and way forward. So Dr. Dinesh and Dr. Neha has already set up the platform about the cholangiocytes, but going into a little brief that cholangiocytes are actually the epithelial cells and they line the intrahepatic and extrahepatic bile ducts. And out of total liver cell mass, it is only 3 to 5% of the cells, which contributes to the multiple diseases of biliary tract. And in humans, if we calculate this whole 3D network of biliary tree is actually 1.25 miles. And that lines the whole bile duct in liver portal tracts and it kind of forms a barrier for any pathogenic attack. If you look at the size, their size is also variable from 16 to 15 micrometer in diameter, but they are one of the long-lived cells in the liver, which has more than 600 days of turnaround time. There are heterogeneity in the cholangiocyte. They are small cholangiocytes and large. Not only they are different on basis of their size, but also in their functionality. The small cholangiocytes are highly proliferative and they have stem-like population, whereas large cholangiocytes are more inflammatory, specialized secretory in function. But they are only 3 to 5% in the liver. Why it is so important? Both bile and cholangiocytes actually maintains the immune defense and homeostasis. And this cholangiocytes actually releases a lot of antimicrobial effect because they form a kind of a barrier to any pathogenic attack. And cholangiocytes protect the biliary tree in general. And similar to gut microbiota, there is a biliary microbiota which maintains the stable physiological microenvironment. So cholangiocytes can also be undergo senescence and apoptosis, which actually influences the immune responses, and due to which and other triggers, it can lead into crosstalk and modulate the immune response, which actually is for bile duct damage and repair and the fibrosis to occur. So, but before they crosstalk with other immune cells, actually cholangiocytes itself are innate immune cells. How? Because they are one of the first responders to any luminal pathogen in the biliary system. They were defined as immune cells because they have specialized receptor present on them, such as toll-like receptor. 2 to 6 has already been defined in humans, and also NLRs, the nod-like receptor, which in fact recognizes the pathogen recognition receptors, which are lined in intrahepatic bile ducts. Not only this, they also secrete antimicrobials to any attack. Say that attack could be from damage, from bacterial invasion, or any other pathogenic attack. So these antimicrobials keep the cholangiocytes in quiescent state. And a huge amount of immunoglobulin A is released by these cholangiocytes. And these antimicrobials plays a big role in keeping the cholangiocytes in an anti-inflammatory state, in a quiescent state. And as I said, they are the first defense. And in release of that, they have mucins, defensins, and other proteins which are involved. In fact, in bile, if you see a second to the albumin, immunoglobulins are higher in number of proteins. Next is their antigen presenting capabilities, which is pretty much debatable because they have both HLA class 1 and class 2, but they do not have class 2 co-stimulatory molecules such as CD80 and 86. But they have this capacity so that they can prime not the conventional T cells, but unconventional T cells such as MATES, mucosal associated invariant T cells, and NK cells because they have CD1 and MR1, which are the receptor for T cells present on cholangiocytes. So if you look at the whole physiology, cholangiocyte itself has the capacity to behave like a immune cells. But whenever there is a insult to the liver, the cholangiocytes gets activated and they can lead to proliferation, fibrosis, and pro-inflammatory secretion. And that is the time when they secrete so many molecules that they invite other immune cells to come and crosstalk with them. This crosstalk can be of three different types. One is direct contact dependent mechanism. Second, they can be bidirectional cytokine chemokine release, which could be paracrine or autocrine, and they can release innate defense molecules. So the major molecules or major cells which are involved in immune crosstalk are monocytes, macrophages, dendritic cells, and also the neutrophils. So there are battery of chemokines, the chemoattractants, which are released by these cholangiocytes.
It's your pointer, Sukriti. The computer pointer.
Yeah, that's So coming back again. So the cholangiocytes, once they get activated, they release battery of chemokines, chemoattractants, and these chemoattractants actually attract other immune cells such as monocytes, macrophages, neutrophils, and these chemoattractants are very specific for the cell type, and these come physically crosstalk or also through other molecules they can crosstalk, as I said. And in turn of that, even they can release the innate defense molecules. So we already know that IL-6, TNF-alpha, CCL2, CD14, CXCL10, they attract the monocytes and macrophages, whereas the damage-associated molecular patterns such as S100, A8, A9, A12, or even metalloproteases, they invite neutrophils whenever these cholangiocytes are activated. And one of the important molecule is IL-6, which is an interleukin-6 that leads to cholangiocyte proliferation, and they work in a autocrine system. So these were the mechanisms which we already know. In non-disease condition, whenever cholangiocytes are activated, they invite other immune cells to crosstalk. But there are new strategies such as extracellular vesicles, which are released by any cell type in the biological fluid, can also carry some of the information which can attract these immune cells and crosstalk with cholangiocytes. So in a steady state, when there is no disease state, it has already been reported by many authors. For example, macrophages, which are one of the key players to crosstalk with the cholangiocytes, whenever there is a microbial antigen invasion, these macrophages, they have a TLR signaling. To keep the cholangiocyte in a steady state, in anti-inflammatory state, TREM2 plays a very huge role. TREM2 is a triggering receptor expressed on myeloid cells too. And this TREM2 keeps the macrophages in a silent state. Whenever this TREM2 goes uncontrolled, there is a huge amount of cytokine storm, which leads to biliary expansion, and that leads to ductal reaction. So whenever it has been done in the preclinical method, where they have shown that TREM2 deficient mice post bile duct ligation developed a more severe phenotype, and as indicated by ductal reaction, and leads to a disease progression. Not only that, if TREM2 is overexpressed, it releases interleukin-33, which is another cytokine, which leads to more of cholangiocyte proliferation. Next, another in the steady state condition, it has been shown in a non-injury model when they gave the bone marrow derived cells in the animal model, they found whenever they gave the bone marrow cells, these bone marrow cells carry macrophages, and these are responsible for the ductal reaction. Now, what this secretes? These macrophages actually secrete a molecule called TWEAK, which is TNF-alpha-like weak inducer of apoptosis, and it is one of the initiating signals for ductal cell proliferation. So keep the cholangiocyte in a silent mode or in a quiescent mode. These molecules were identified as a important key players. But whenever there is a disease condition, then what happens? What is the clinical relevance of these crosstalk in various diseases? So I will start with the MASLD. This is a very recent paper in Gut by Liu et al., where they have done is a transcriptomic analysis of both human and mouse liver using the multiplex immunofluorescence, also biliary on-chip model and some knockout model. They have identified whenever there is an activated cholangiocytes, it induces the acute phase protein, which is ORM2, and that leads to ductular reactions in both human as well as mouse livers. So this ORM2, they have confirmed with a unique transcriptomics, which actually leads to changes in the macrophages, liver macrophages through calcium channels. And these macrophages leads to a pro-inflammatory state. So this ORM2 has been identified both in liver of mouse and human to understand the crosstalk between cholangiocytes and the macrophages, which plays a key role in pro-inflammatory state. The other very interesting study published in Nature Medicine by Prakash Ramachandran also, that they identified 940 MASLD patients where they checked all the complete spectra, and they created a secure, searchable, open resource, which is SteoSite. And this, including the histopathological assessment, as well as their transcriptional data, they have formed a 15-gene transcriptional risk score to predict the future of hepatic decompensation events. This is also in MASLD. And they found that these ductular reaction cells has a huge number of CD68 macrophages, which is one of the key players in tissue and periportal area. So this already been confirmed in other papers where they have found that ductular reaction cells actually, which leads to more of fibrosis and portal inflammation, has increased amount of CD68 macrophages and also CD8 lymphocytes. And these not only leads to the progression, but also inflammatory state, necrosis, and further deterioration of the disease. The other molecule is osteopontin, which has already been identified that it is released from the cholangiocytes, which acts as a chemokine to the macrophage recruitment. So other than the battery of chemokines, which I have mentioned earlier in my slide, the osteopontin also plays a huge role. So osteopontin actually has a XCL19, which induces osteopontin secretion, which programs the macrophages and leads to activation of the hepatic stellate cells. So they have shown in the normal liver and MASH, the osteopontin along with the ductular reactive cells, their expression increases. And in the advanced MASH condition, this linearly increases all the chemokines. So this plays a huge role as well. This is a recent paper in Liver International. So this was about macrophages, but mast cells also plays a important role, which has been recently identified in a preclinical model where they have given a high-fat diet. Whenever we give a high-fat diet, there is a biliary senescence, which we already know. And this biliary senescence cells, which are present in the ductular reactive cells, release lot of insulin growth factor 1, IGF-1. And this IGF-1 receptors are present on the mast cells, which leads to the inflammation and fibrosis. So mast cells has been identified as one of the key players for the ductular reactive cells. So there's another publication which linearly shows that how these mast cells are changing from normal to NASH end stage. They were identified during the NASH where mast cells get activated. But what is the mechanism? So these mast cells actually releases histamines. And this has been shown that when whosoever patients take histamine H2R/HR antagonists have a lower prevalence of NAFLD. And these receptors are present on the cholangiocytes, which via VEGF activates the large cholangiocyte proliferation and leads to liver fibrosis and inflammatory response. So there is a huge link between the biliary-derived histamine and NAFLD progression through the receptor which are present on the cholangiocytes. So other is also another important study in alcoholics. So whenever we talk about NAFLD, it was more macrophages which were crosstalk. In alcohol, we know neutrophil number also increases, and this has been shown in hepatology paper where they have found that degree of ductal reaction expansion actually correlates with disease progression and short-term survival. And they found that the cytokeratin positive KRT7 cells, which are higher in number, and their gene expression profile also increases. And they found that all the chemokines which were present in this KRT7 sorted cells were actually attracting neutrophils. And histologically also, they have shown that neutrophils infiltration is higher in alcoholics at the periportal area. And using the liver organoids also, this neutrophil migration, enhanced cytokine expression was also identified. Even in another study, which is published in Gut, clinical and histological molecular profiling of different stages of alcohol-related liver disease in non-decompensated ALD and compensated ALD alcohol patients, they have also identified huge number of infiltration of neutrophils which were present in the ductal reactive cells. So one of the key players, which I will skip for now, but gut microbiota also plays a huge role because we know that gut changes its abundance of bacteria, and that leads to dysbiosis. And this dysbiosis triggers the cholangiocyte proliferation, which leads to more of neutrophil infiltration. This is still in infancy, and we have to work more in details about this. So now coming to the viral hepatitis. So there were very few publications which showed that cholangiocytes has some kind of a role in viral hepatitis. But in fact, very interesting finding which I found in one of the older papers in 2015, where they have shown, because we know the origin of cholangiocytes and hepatocytes are same, but still, cholangiocytes do not express all the receptors which are involved in the viral entry for HCV or HBV. But one of the publications showed in the in vitro condition where they have found that cholangiocarcinoma derived cell lines, they carry HCV entry receptors present on them, and they are also involved in the replication. But still, the primary cholangiocyte lacked the expression of entry factors. So could be during the cholangiocarcinoma, these receptors are expressed on these cholangiocytes, which are involved in the further aggression of the disease. And two of these cholangiocarcinoma lines supports this efficient entry and full virus replication. Not only that, one of the older publication also showed that hepatitis B surface and core antigens in the cholangiocytes are present in the minority of the cases. So that could be one of the indicative that the patients who are progressing to the disease may express these cholangiocyte receptors, which are involved in the viral entry. In fact, one more publication which I found where they have shown that during the cholangiocarcinoma, these hepatic progenitor cells and cholangiocytes crosstalk leads to more of inflammatory cell activation, and that leads to more of fibrosis, which leads to cholangiocarcinoma. As I said, that cholangiocytes also have mucosal associated invariant T cell receptor present on them. So this publication showed that whenever there is a mate cells in cholangiocarcinoma, they are heterogeneously depleted. And in the patients where there depletion of these mate cells, there is an increased bacterial burden within the tumors, and the high presence of mate cells in liver or cholangiocarcinoma predicted long-term survival in two independent cohorts. So the mate cell number in ductal reaction cells are actually good. And if they are depleted, those patients has a lesser survival. Then we come to the cholangiopathies such as PSC and PBC. This is very well defined, and in the consecutive cholangiocyte lecture series, people will cover these in more detail. But to give you a brief, when in the PSC, when they did the experimental pipeline and using the spatial analysis, they identified few of the immune cells which were highly present in PSC and leads to the inflammatory state. And they found that phagocytic mononuclear cells as well as neutrophils from innate immune cells and Th17, CD4 and CD8 T cells are actually present more close to the bile duct within the fibrotic areas, and they modified the number, location and function can lead to a progressive PSC. And a recent abstract by Nidhi from Mayo Clinic in ASLD 2025, where they have identified whenever there is a trigger of cholangiocyte through stress or other reasons, the etiology we do not know yet, but this stress actually leads to more of DNA damage, which releases the C gas and STING pathway, which recruits more of neutrophils and leads to oxidative stress and inflammatory and fibrosis. This infiltration actually propagates the oxidative stress and perpetuates the biliary fibrosis and inflammation. So C-gas STING pathway is also one of the identifying features of the PSC. So there is a interplay of innate immune cells in PBC because whenever there is a trigger, there are short-lived immune cells. The short-lived immune cells, they are recruited from outside, like macrophages, monocytes, dendritic cells. But whenever there is an uncontrolled inflammatory state, they releases their battery of chemokines, cytokines, and trigger the resident immune cells, which are innate lymphoid cells or even endothelial cells, Kupffer cells, which actually releases more of cytokines and antimicrobials in the PBC, which has been defined. So there are many bile acid receptors which are present on cholangiocytes. So TGR5 and FXR, which will be in detail covered by Dr. Omkar. But to give you a brief, in PBC, they have been defined that these bile acid carry bacterial products or other triggers or DNA damage proteins, which can trigger these cholangiocyte present bile acid receptor, which counterbalance the pro-inflammatory TLR signaling. And this maintains the mucosal homeostasis and the metabolic regulation. So many treatments are being targeted for these FXR and TGR5. This TGR5 actually leads to activation of cholangiocytes and invites more of inflammatory macrophages and neutrophils to the site of ductal reaction. So if I summarize the cholangiocyte immune crosstalk relevance, in MASLD, we have to remember that there are more of CD68 positive macrophages, which expands into the central lobular region. And the degree of these CD68 number of function can lead to extent of portal inflammation. And one of the leading molecule, ORM2 plasma levels, can also help us identify the ductal reaction. Whereas in alcoholic hepatitis, it is more of neutrophils which are expressing chemokines and recruited neutrophils to the periportal area through cholangiocyte. Whereas in viral hepatitis, it is the more of ductal reaction leads to extent of severity of the disease. In PSC, it is neutrophils, Th17 and phagocytosis. In PBC, it is C-gas and oxidative stress. Whereas in cholangiocarcinoma, there is a huge ductal reaction which is directly proportional to the tumor size and the degree of inflammation. So besides chemokines and immune cells, interestingly, recently by Gregory Gore's group, they have found that cholangiocytes can release extracellular vesicles, which can crosstalk with the macrophages. To give you a brief, extracellular vesicles are small vesicles which are derived from cell, and they are miniature of the cell, and they carry all the information of the cell inside them in form of cargos. So they identified that these activated cholangiocytes, whenever there is a trigger, they releases these EVs, and these EVs contain damage-associated molecules such as S100A11, where receptor such as RAGE is present on the macrophage. They identify this RAGE and leads to more of inflammatory pathway and release TNF alpha, IL-1 beta. So these extracellular vesicles can also aid in the crosstalk. So once we found that this is the thing that they can have another type of a crosstalk, even in ILBS, we have investigated extracellular vesicles. So what we have done is we have recruited 15 PBC patients and 90 autoimmune hepatitis patients because they have a lot of pathological and clinical features which are similar. So we took two cohorts and compared them with healthy controls. We took their plasma as well as the stool and fractionated both bacterial as well as the host extracellular vesicles, identified them phenotypically, identified their bacterial origin, cargoes, and their associated protein cargoes, and further virulence factors. So this is the identification and the fractionation of the plasma, extracellular vesicle, as well as stools. This is the Western blot, where we can see that the 10 to 16 fractions are actually the host extracellular vesicles, whereas the bacteria are the earlier fractions. And the transmission electron microscopy identified that these small vesicles are actually extracellular vesicles. So once we identified these EVs, we did the 16S metagenomic to know whether there is a difference in alpha and beta diversity. To our surprise, beta diversity of PBC and AIH were absolutely different. And we found that acetobacteria producing extracellular vesicles were significantly lower in PBC, whereas it is higher in AIH. So we can use acetobacteria as an identifying molecule for the PBC. And also these virulence factors were identified on these AIH and PBC plasma extracellular vesicles. And we found virulence factors, which is VGF0161, which was associated with Pseudomonas and Streptococcus. These virulence factors are present, which are associated with highly in PBC than in healthy and AIH. Not only this virulence factor were also has a mimicry epitope, which is from the host, which is pyruvate dehydrogenase complex E2, and we have identified a peptide which could trigger these triggers in the PBC as well as in autoimmune disorders. So when we looked at the protein cargoes of the extracellular vesicles in PBC, this was also different between mild, moderate autoimmune and PBC. If you look at the expression profile, this is healthy, this is PBC, mild and moderate. This is absolutely different cargoes. So we thought, can we make a panel of cargoes of EVs which can differentiate between PBC and autoimmune? So yes, we could see that we can differentiate severe AIH with PBC with two of the proteins, which are spectrin beta chain and serum peroxidase, which are different in AIH and PBC. Whereas if we have to differentiate between mild autoimmune and PBC, immunoglobulin heavy chain variable A, kappa, and vitronectin can be used as a panel to differentiate between PBC and AIH. To summarize this study, we have developed a novel method to fractionate bacterial vesicles and EVs from both plasma and AIH. So total EVs are elevated in both AIH and plasma, but their cargoes are different in two populations. And the plasma EVs have altered bacterial origin and they carry different virulence factor. And we can design or validate our panels to differentiate PBC on basis of EV protein cargoes. So, but there are many unresolved questions such as cholangiocyte, how it switches from anti-inflammatory state to inflammatory. Can this immune niche be rewired for therapeutic purposes? And this 3D tissue organization crosstalk can be investigated. So the way forward could be, which we can design studies on, the high throughput immune niche mapping through spatial omics or machine learning to be developed to crosstalk phenotype and patient outcomes, or Dr. Dinesh, as he's doing the organoid system, so we can do the immune crosstalk in these co-culture and preclinical testing. And there could be more clinical trials of rationally designed multi-targeted approach. So thank you. I would like to thank my mentor, collaborators, and the team. And thank you for your attention.
Very nice, Sukriti. Very good talk. And I see very few PhD students here. It should be mandatory for them. And also, they missed a lot. So, my 2 points is the mast cell picture, which you showed, also showed the reduction in the size of the cholangiole. Uh, and it does it mean the mast cell is destructive to the uh,
Yes, sir.
cholangiocytes? So when the people have itching, I thought itching was a very protective thing. Here you show that there is a reduction in the size. So mast cells are destructive.
Yes, sir. Actually, that's why there are studies which shows that who are on antagonists of histamines, they are protective for NAFLD. So mast cells are actually releasing huge amount of histamines during liver injury, and those receptors are present on the cholangiocytes, and those receptors actually leads to pro-inflammatory condition.
So there is something called as mastitis. Mast cells are increased, and that was one area. And second, it is very interesting that you showed autoimmune versus PBC. It would be worthwhile to see the effect of steroids or treatment and what happens to the EVs. So, can EVs be used as a therapeutic target for success of autoimmune or PBC therapies? Can be looked into. Any other comments? Well, thank you. Very exciting.
Thank you so much.
Sorry. Sorry for that. So, good morning, and thank you, Sukriti ma'am, for that enlightening talk. So, the topic given to me today is the benefits and challenges in targeting bile acid circulation in cholestatic liver disease. And I hope to discuss this topic along the following outline. Why bile acids? Why now? The fundamental physiology, the benefits of targeting bile acid circulation, therapeutic strategies, targets and challenges, future directions, and then a quick summary. So, why bile acids? So the hype around bile acids is not new, but it is definitely renewed. And this is primarily because of a better understanding of the mechanisms and the development of therapeutic targets. So cholestatic diseases cholestatic injury is driven by toxic bile acids, and treatments have always been limited. UDCA has been around for about 20 years, and newer therapies have been available only for the past 10 to 12 years. Even pediatric cholestatic diseases such as PFIC and Alagils now have multiple pharmacological therapies, which can avoid surgical diversion. So what is the enterohepatic circulation? So you have cholesterol, which is present in the liver, and from this cholesterol, there is formation of bile acids through the rate-limiting enzyme CYP7A1. These bile acids, once they are formed, are conjugated with either glycine or taurine. These primary bile acids include cholic acid and chenodeoxycholic acid. These are ultimately amphipathic molecules which have a detergent effect. They have both a hydrophilic and a hydrophobic component. These conjugated bile acids then are subsequently through the BSEP and MRP secreted into the bile canaliculus into the small intestine. Over here in the small intestine, it helps through its detergent effect in absorption of lipids. As it moves through the intestine, due to microbial action of bile acids, these are converted to secondary bile acids, which include the deoxycholic acid and the lithocholic acid. These further move down and they reach the terminal ileum where they are reabsorbed into the blood. More than 95% is reabsorbed through the ASBT. These apical sodium dependent bile acid transporters, which are present on the luminal side, take it up, and through the OST alpha beta, this is transported to the blood. Once it reaches the blood, it goes into the portal circulation where it is taken up by the liver through the NTCP and OATP1. Around 5% of these bile acids move further down into the colon, and of these 5%, 70% are reabsorbed, and barely 5% is actually excreted in the feces. Now, once
Your pointer.
sorry, sir. So, once these bile acids are present in the blood, and those which escape the first pass metabolism, the first pass clearance, the non-protein bound bile acids are also filtered by the liver. Now, by the kidney. The kidney also has presence of ASBT receptors, which is present in the proximal tubules. Now, reabsorption from here also helps in bringing back the bile acid pool into the blood. This is important because there are theories which suggest that presence of bile acid in the urine may cause injury to this renal proximal tubular cell, which can further cause injury to the distal tubular cells, cause sloughing, and ultimately lead to cholemic nephropathy. So now in cases where the bile acid load is very high, the bile acids are secreted from the liver into the blood via the MRP3, MRP4, and OST alpha beta, which leads to systemic complications. There is also presence of an FXR receptor, which is a nuclear receptor. Now, this FXR receptors are present in two places, predominantly the hepatocyte and the ileal enterocytes. In the hepatocytes, the bile acids activate the nuclear receptor FXR, which leads to formation of a small heterodimer molecule, which ultimately results in inhibition of CYP7A1 and reduces formation of bile acids. These FXR also help in upregulation of BSEP and MRP2 and help in canalicular excretion of bile acids. The presence of FXR in ileal enterocytes also has a role. Now, stimulation of FXR in ileoenterocytes causes release of FGF19, which further stimulates the FGFR4 on the hepatocytes, and that causes downregulation of the CYP7A1 and reduces bile acid synthesis. TGR5, like we've also discussed, also has a role in the enterohepatic circulation. Now, TGR5 is present on multiple tissues all throughout the body. Most importantly for us is the intestine. Now, stimulation of TGR5 by bile acids, especially the lithocholic acid, causes increase in the GLP-1 secretion. Now, increase in GLP-1 helps in glycemic control and also reduces the satiety, increases the satiety and reduces the gut movement. In the pancreas, there is also this release causes increased insulin sensitivity and also reduces the improves the glycemic control. In the gallbladder, it leads to gallbladder refilling and helps in bile acid hemostasis. And in the macrophages, it is shown to have anti-inflammatory, anti-atherosclerotic properties. In the muscle, muscle and the adipose tissue, it leads to thermogenesis and regulation of the body temperature. So what are the benefits of targeting bile acid circulation? So what are the functions of bile acid? We'll briefly discuss this. We already know that bile acids have a detergent function and they help in absorption of fat, but they are also playing a role in different organs. In the liver, it helps in the lipid and bile acid metabolism. In the pancreas, as we've discussed, it increases insulin secretion and improves the insulin sensitivity. In the adipose tissue, it helps in thermogenesis, which is along with the thyroid receptor hormones. In the immune system, it has an immunomodulatory effect. And in the brain, it has a neuroprotective effect. Studies have shown that increased bile acids within the brain can cause neurodegeneration and neuroinflammation. So what goes wrong in cholestasis? Like we have seen, now bile acids affect multiple organs, but what happens in each organ? So in the liver, there is the bicarbonate umbrella, which is predominantly secreted by the AE2 cells, where chloride is exchanged for bicarbonate. Now this bicarbonate umbrella plays a protective role, and it protects the liver and the cholangiocytes from toxic injury from the bile acids. This AE2 is CAMP mediated, and in cases of inflammation, there is downregulation, which causes increased injury. The cell membranes within any organ has a lipid bilayer. Now, presence of bile acids in a toxic quantity causes a detergent action even on this lipid bilayer and causes increased permeability. Following this, the bile acids can seep into the cells and cause injury. This leads to subsequently mitochondrial injury, ER stress, hepatocyte and cholangiocyte injury. As discussed, increased filtration may cause increased propensity of cholemic nephropathy. In the brain, this may cause neuroinflammation and neurodegeneration. In the skin, this causes pruritus, which is what most of our patients present with. And this can also ultimately cause sarcopenia because of increased quantity of inflammatory cytokines. So blocking these targets would help in improving patient outcomes. So what are the therapeutic strategies, targets, and challenges? So there are multiple targets like we've discussed. And these include most commonly are the targets which include the cholhepatic shunting, which is UDCA, nor UDCA. There are targets which affect, another target is ASBT inhibitors, which reduces the bile acid uptake, increased fecal excretion. FXR, which again reduces bile acid synthesis, increases secretion. FGF15, which acts along the lines of FXR. And also we have the PPARs, which help in reducing bile acid synthesis, increase bile acid glucuronidation, and bile acid secretion. Also, TGR5, like we discussed, is present on multiple cells and may also be a beneficial target. NTCP is another one, not labeled here, but NTCP also, which helps in uptake of bile acids, is a therapeutic target for cholestasis. So UDCA, this is a tertiary bile acid and it comprises of about 1 to 3% of the bile acid pool. This is a very well-known molecule and its mechanisms include detoxification of the bile acid pool by increasing the hydrophilic bile acids and therefore competing with the hydrophobic ones. It also has a cholecteric effect which causes an increase in the bile flow. It also has cytoprotective and anti-apoptotic effects by stabilizing the mitochondrial membranes and reducing the reactive oxygen species. It is also immunomodulatory and has an anti-cholesterol effect by reducing the cholesterol secretion into bile and therefore reducing the prevalence or helping in dissolved gallstones. So we have been using UDCA now for almost 20 to 25 years, and the most common indications would include PBC, PSC, and ICP. Many studies have shown a long-term benefit with PBC, with improving the overall survival and the transplant-free survival. Problem is that about 40% of patients do not respond to UDCA. When you talk about PSC, there is not a lot of benefit of PSC, at least in terms of long-term patient outcomes. UDCA does improve the enzymes, can reduce ALP, but no studies have shown that it improves survival, it improves the histology, or prevents cholangiocarcinoma. A recent study was done which evaluated high-dose UDCA, which found actually an increased mortality. Apart from that, you have the intrahepatic cholestasis of pregnancy. Now, UDCA in ICP has shown an improved or better survival of the fetus and improved symptoms, that is reduced pruritus. When we talk about norbucolic acid, this is previously called as NOR-UDCA. This is actually a truncated form of UDCA, which actually lacks a methyl group. Now, because of this NOR-UDCA is resistant to conjugation, and this ultimately undergoes multiple cycles of cholhepatic shunting. Cholhepatic shunting is when bile that is secreted into the cholangiocytes are reabsorbed and via the peribiliary plexus again go back to the hepatocyte. This subsequently then undergoes glucuronidation and elimination. This cholhepatic shunting causes bicarbonate-rich cholerasis. In mouse models, this has been shown to have anti-cholestatic, anti-inflammatory, and anti-fibrotic properties. Now, NOR-UDCA, because of these properties, has been evaluated in PBC and MAFLD. This one of the first studies was in 2017, which was a multi-center phase 2 study, which included 161 patients who received NOR-UDCA at doses of 500,000 or 1500. This was compared to a placebo. They found that as compared to placebo, NOR-UDCA significantly reduced the ALP levels, and it was well tolerated. The side effects in this study included nasopharyngitis, pruritus, fatigue, and sometimes headache. Interestingly, though, that once this NOR-UDCA was discontinued at week 12 and followed up for another 4 weeks, there was a rebound increase in the ALP. So this is actually the NUC5 trial is a phase 3 trial. This is yet to be published, which has included 301 patients with PSC. And these patients received 192 weeks of 1500 milligrams of NOR-UDCA or placebo. Their primary endpoints included ALP reduction or disease progression, which was histology based. At 96 weeks, the interim analysis shows that there is partial normalization of ALP with no worsening on histology. Another study has evaluated NOR-UDCA for MAFLD, and this phase 2 trial included 198 patients. These patients were given 500 milligrams of NOR-UDCA or 1500 versus placebo for a total duration of 12 weeks. They included patients with a clinical diagnosis of MAFLD who had an elevated ALT. Their primary objective was a mean reduction in the ALT concentrations. So now when you look at this, NOR-UDCA in 500 and 1500 milligrams cause a significant reduction in ALT levels as compared to placebo. Now, NOR-UDCA's role in MAFLD is primarily likely because of presence of microcholestasis. So in MAFLD, there is a change in the composition of bile acids with altered FXR FGF19 signaling, which causes increased CYP7A1 expression, and this can lead to impaired hepatobiliary functioning and hepatobiliary transport functions. All these lead to microcholestasis. In India, very recently, based on a phase 3 trial which is yet to be published, NOR-UDCA has been approved for MAFLD. This study has shown a normalization of ALT and a reduction of fibrosis. Although this studies did utilize only fibroscan and biopsies were not done. So the challenges with NOR-UDCA primarily include that it is a new molecule and we do not have any long-term data on the safety. Also, we do not have any data on the hard clinical endpoints such as overall survival or transplant-free survival. Next, we come to the gut-restricted ASBT inhibitors. As we've discussed, ASBT is present in the ileal enterocytes and in the kidney, which helps in reuptake of the bile acid. So one of the first studies which was done was the ICONIC trial in which they used maralixibat for Alagille syndrome. In this phase 2b study, they found that maralixibat improved the itch score by 1.6, by a median of 1.6, and also reduced the serum bile acids by a median of 96 micromole per liter. The ICHRO score is basically a 4-point score, which is similar to the VAS score which we use, which is a 10-point score. Following this, FDA approval was given for maralixibat for Alagille syndrome. Similarly, in 2023, this was used for PFIC, and this also found a reduction in bile acids as compared to placebo. In 2024, this was approved for PFIC. Then odevixibat is another ASBTI molecule, which is again being utilized for PFIC. Two studies have been done. One is a phase 3 trial and another extended trial. And both of these have found that there is a 43 to 58% reduction in the pruritus with a marked reduction in the serum bile acids. Based on this, based on the PETFIC 1 and 2, approval was given in 2021. A recent 2025 study also showed that almost close to 73% of the patients who were on odevixibat showed symptomatic improvement. They also found a cutoff of bile acids of more than 333, being a marker of poor response. When we talk about adults, maralixibat has been assessed in PSC and PBC in adults, but the heterogeneity in the data and the heterogeneity in the response do not give us any conclusive results. Linerixibat has been utilized in PBC. There is a phase 2b and 3 trial, and this has shown that there is a dose response of itch in patients, and with a dose of 40 milligrams twice a day, the pruritus has significantly improved. So this is currently undergoing FDA evaluation for approval for PBC. So apart from that, this linerixibat has also shown improvement in sleep in these patients. So apart from talking about the gut-restricted ASBT inhibitors, we also have the systemic ASBT inhibitors. So these molecules can target both the ileal enterocytes uh, and the ASBT receptors in the proximal renal tubule. So this systemic ASBT inhibitors has a theoretical risk of worsening cholemic nephropathy, like this as we discussed, because this can cause detergent action and then ultimately sloughing and lead to bile cast nephropathy. But there are two mouse models which have been performed in 2024 and 2025, which show otherwise. These studies have shown that by giving the systemic ASBTI, there is a reduced proximal tubular injury biomarker such as KIM1, along with lesser tissue damage and lesser fibrosis. The only point to consider is that both these studies in mouse models have been done by the same group, and reproducibility is to be looked at. Another systemic ASBT inhibitor is A3907. So this was orally administered in a bile duct ligated model. And this found that using this inhibitor causes increased urinary bile acid elimination, reduced serum bile acids, and improved markers of liver injury. They had also utilized this as a phase 1, and this was given in healthy human subjects for 7 days as a daily oral administration. They found that when they evaluated the serum bile acid levels, it was significantly lower in the postprandial period, which you would expect in these patients. So therefore, the molecule was acting at the ileal enterocytes. But the dose that was required, the dose that was given in mice, and the modified dose based on the weight which was given in adults, it showed that the systemic exposure was several fold lower, which means this likely has a very poor absorption and does not translate yet to clinical use. So the challenges with ASBT include that the predominant molecules that we have are gut restricted, and most of them develop GI side effects such as diarrhea and abdominal pain. And these would be ineffective in those with a severe block in bile acid secretion, since none of it would reach the ileum. And although these drugs show an improvement in symptoms, again there is no data on hard clinical endpoints such as event-free survival. Systemic ASBT are currently unavailable, and a small molecule IBAT inhibitor with acceptable bioavailability, pharmacokinetics, and renal tubular target engagement is yet to be discovered. So next we talk about the FXR agonist, which is the next therapeutic target. So the first trial in this was the POISE trial, in which they had evaluated obeticholic acid in 2016, in which they found that a significantly higher composite response compared to placebo. 46% of those who are on obeticholic acid responded as compared to placebo, with sustained normalization of ALP and GGT. Although this study did not show any change in the fibrosis. Based on this POISE trial, this was given accelerated approval as a second line in PBC. Subsequently, a 2025 publication, that is the phase 4 COBALT trial, demonstrated concerns of overall efficacy, and therefore this drug has now been withdrawn from the market, from the European market. Other drugs that included are the Cilofexor, which was the initial trial was done in 2019, which was a phase 2 trial. And this evaluated the change in ALP levels from baseline and found that usage of Cilofexor in patients of PSC causes significantly reduced ALP levels as compared to the placebo. But a subsequent study, which was done in 2025, the PRIMUS trial, which evaluated histological progression in PSC on Cilofexor, was terminated prematurely after just enrolling 160 patients. This is because at 96 weeks, this showed a 6.8 probability of detecting a significant difference. Their cutoff of futility was 10%. So there's also the REGENERATE trial, which has evaluated obeticholic acid in patients of MASH with advanced fibrosis. And this has found that obeticholic acid increased the proportion of patients who had a more than one stage fibrosis improvement as compared to placebo. So 23% of patients on obeticholic acid had more than one stage fibrosis improvement. Almost 6.5% had NASH resolution without worsening in fibrosis in the REGENERATE trial. Now, the primary reason this has not yet received approval is because of the adverse effects of pruritus and elevated LDL levels. This elevation in LDL level because of FXR agonists is predominantly mediated by the SERP pathway and a reduction in LDL receptors on the hepatocytes. So the challenges with FXR agonists is likely that FXR is already highly stimulated because of the presence of retained bile acids, and its stimulation is it cannot be stimulated further because it is maximally repressed. If we do develop agonists, we require agonists which have a very high affinity for these receptors. Obeticholic acid has been shown to cause pruritus and elevated LDL levels, and therefore would not be helpful in patients of MAFLD. And the side effect profile of non-steroidal FXR agonists such as cilofexor are still yet to be evaluated. So along the same lines of the FXR agonist, we have another molecule, which is the FGF19 analog. Now, just to recall, FGF19 analogs, they cause a stimulation of the FGFR4 on the hepatocytes, which cause reduced CYP7A1 expression and therefore cause reduced bile acid formation. So this phase 2b placebo-controlled trial, which included 160 patients over 48 weeks, evaluated the primary endpoint as changes in ELF. Now, these patients were dosed at 1 mg and 3 milligrams of aldafermin, and they found that at 3 milligrams, there was a significant reduction in the ELF as compared to placebo. This was not seen in the group which received 1 milligram. Although these changes were noted in non-invasive markers such as ELF, histological changes were not significant. The next class of drugs for us is the PPAR agonists, and these include the benzafibrate, fenofibrate, seladelpar, saroglitazar, and elafibranor. So the first drug was actually benzofibrate, which showed that there was a 31% response as compared to no response in the placebo arm in patients of PBC. And this benzafibrate was also then subsequently evaluated in the FITCH trial, which showed a symptomatic reduction in the pruritus in these patients. Of note or of importance to us is the EPICS trial, which was initially conducted in 2021, which was initially a phase 2 trial of saroglitazar in patients of PBC. Over here, they evaluated 2 and 4 milligrams, and they found that there was a sustained ALP reduction in about 50% of the patients who received saroglitazar as compared to 3% with placebo. There is a phase 3 trial, which was data of which has been shown in August 25, that is the EPICS 3, but it is yet unpublished. That also showed good response to saroglitazar, although the doses used in that was 1 and 2 milligrams. Apart from that, there is a response trial which was conducted in 2024, which evaluated seladelpar in patients of PBC who had an inadequate response or non-response or were intolerant to UDCA. This drug was given for 12 months, and they found that with seladelpar, the response rates were as high as 61% as compared to 20% in placebo. ALP normalization and pruritus also improved in this population. Based on this, seladelpar received FDA approval in 2024. Another trial is the ELATIVE trial, which evaluated elafibranor, again for PBC. Over here also with elafibranor, there was a significant response and ALP reduction, 51% as compared to 4%, with additional improvements in the GGT and inflammatory markers. Although this trial did not show any differences in pruritus. It would be noteworthy to say that both the response and the ELATIVE trial both do not have long-term data on event-free survival. So the challenges with PPAR agonists include that these can cause mild transient transaminitis, which spontaneously resolve. It can cause myalgia, elevated creatinine, and PPAR deltas can also cause a weight gain. Again, as discussed, long-term safety data is currently unavailable. Other therapeutic targets would include the NTCP inhibitors. Now, NTCP helps in reuptake of bile acids. NTCP inhibitors include bulevirtide. Now, this is also a molecule which prevents hepatitis B and HDV from entering the cell. Studies on Hep B and HDV have shown that patients who receive bulevirtide also showed an increase in serum bile acid, and this remains a therapeutic target to be further investigated. TGR5 inhibitors are only investigational, and there are preclinical trials which show some response. So future directions, we need to evaluate combination therapies in in this group of patients, and we need to develop absorbable targeted systemic ASBT, which can also target the renal ASBT receptors. We require development of TGR5 selective agonists, which can help in organ protective effects. Diet modification is also an area which needs to be looked at, because studies have shown that insoluble fiber can bind and sequester cholesterol and the bile acids in the intestinal lumen. Further, evaluation of FMT as an intervention needs to be assessed. One study by Jasmohan Bajaj had shown that presence of lower secondary bile acids post FMT was a marker of non-response of FMT to FMT for HE. So to summarize, multiple therapies are available in our armamentarium today. All are early stage and long-term data is awaited. Few drugs in the pipeline for approval, such as linerixibat and saroglitazar for PBC. And there is a need for better therapeutic targets in PSC. As we have seen, there are barely any molecules that we can use today. And a deep dive into the nephrohepatic circulation may provide newer targets for treatment. Thank you.
Very well covered, Onkar. We could have had more questions and answers, but we have today a very good day for ILBS. We have new entrants for PDCC. I see all of them here. Are you all here? You're welcome to ILBS. We hope all of you can join us, but I know some will have to probably wait. With that, I think we have PDCC organ transplant anesthesia, PDCC intervention radiology, hepatopathology, advanced diagnostics, clinical microbiology, gastroradiology, and cellular transplant immunology, diagnostic virology, and dialysis therapy. Well, welcome all of you. I do hope you have a good day, and I would like to meet you before you leave after your. Thank you.